Good morning, everybody, and thanks for joining us at this, at least in California time, early hour. And I've been asked to reintroduce myself. So my name's Henning Hermjakob. I'm coordinating the proteomics services team at the European Bioinformatics Institute in Cambridge, UK. And we are a partner on the UCLA BD2K Center of Excellence. And I'm going to present one of the three projects we are working on right now in the context of this grant, and that is the Omics Discovery Index. For those in the room here, I'm afraid the first few slides will be just a repetition from yesterday, but I need to give the introduction on background, so I'll try to go through these quite quickly. So the vision of the Omics Discovery Index really is to come to achieve what PubMed has achieved for publications, which is one central entry point for searching all the relevant literature. And in publications, this is fairly straightforward. For data sets, this is very far from straightforward. Most data sets which are available nowadays publicly, even if they are in repositories, in well-structured repositories, they are available mostly in technology-specific repositories, and you have many different entry points to search for relevant data sets. If you want to look across multiple omics technologies, you have to put the same query, but very likely in different query languages or encodings to the different resources. And even Google doesn't work well because you can't targeted search for data sets in Google. And so really we want to achieve a similar structure like what PubMed has done for publications for omics. So the specific aims are to provide a data discovery index for genomics, proteins, to integrate across resources on both sides of the Atlantic, to integrate open and controlled access resources. Our test case for the controlled access resources is the EGA, which is the European counterpart to dbGaP, and which publishes the metadata of studies, but it does not, for obvious reasons, the actual data, and you only identify what you're interested in, and then you have to go through approvals to get access to the actual data. I'm looking, if I'm hesitating from time to time, then this is because I'm looking at the monitor I have here to see when the actual NIH, the webinar has updated the screen. So we set up one computer so that I can see that update. So I'm trying to keep both in sync and manage the delay. So the idea is to provide the, to also from the beginning build the system up so that we can easily extend it, that we can easily add new databases and repositories into the already existing federated structure with reasonable effort. So on the next slide, we'll show the outline structure of the system as soon as it shows up. And we are implementing a federated approach. We are asked all the participating resources currently from proteomics, metabolomics, and genomics provide for each study they contain, for each data set they contain, a specifically formatted short XML metadata message, which is then collected and jointly indexed by the OmicsDI system. The OmicsDI backend is using the existing EBI indexing infrastructure based on Lucene, and this is a full production system at the EBI, which is currently indexing more or less all the data content of the EBI. So something like more than 1 billion entries. And so we are fairly confident that it will scale up for the needs of this project. And also the scale, building on this existing infrastructure gave us the chance to really get a running start, and rather than investing lots of time into building up the backend infrastructure, we could just build on what is already there, and also integrate what this project had learned already previously. But we, of course, had some work, and we had to build the actual web application, the presentation over the web. Everything is completely decoupled between back-end and front-end through web services. And we also added one additional component, a database component, to be able to collect access metrics. So every access through the data, through the portal, will allow to be counted, and so we can provide specific dataset-specific access metrics. Going a bit more into detail, the XML we are collecting has, for obvious reasons, because we are going across multiple different omics types, has a selection of fields, a core set which is mandatory, then a set of desired fields, and then there can be lots of additional fields, which tend to be the technology-specific ones. And to support the easy integration and reasonably efficient integration of new resources, we have added recently a validator step so that we can easily automatically evaluate whether the provided XML is valid, and the validator then reports for each file it provides different warning levels between error and warning to point out formatting errors. So most of what will come up on the next slide in a second in the webinar system, I've already said. So I'm going to skip over that one and move right on to the next one. One advantage of using an existing indexing system is that we can, if we want, we can easily tap into the richness of the underlying infrastructure. And for example, for proteomics, we can index for a given data set, not only the metadata, but also the identified proteins. And we can then, if we want, easily link out to all the EBI resources on these proteins. This is not implemented in the current resource, but of course, because we tag into the existing system, that is fairly straightforward to do. Okay, I'll jump over the next slide, which again just the slide called DDI application just gives a brief shows those components we had to develop for the web interface, but you will see these in practice in a minute, so I'll skip. The current status as of August of the project is that we have a publicly available alpha release, which indexes 6 repositories from 4 organizations across 3 omics types and 2 continents, with altogether about 5,000 records. We are currently in the stakeholder feedback phase. We have shown the alpha to a number of stakeholders, and once we have closed this phase, we will go into the next development iteration and release a full beta version latest in January next year, but likely significantly earlier. This is just a brief overview, just as background to, because the question came up yesterday on the stakeholders we have discussed with, where we have given presentations. The major one after this one is a BioCaddy webinar, which is planned for the 28th of August. This still needs to be confirmed, but it looks likely to happen at that date now, currently. Okay, and with that, I will move into the demo. So this is the live version of the tool, and in the first row of panels here, you see overviews of the current content of the indexed data set resources. There's a Wordle which is generated from all the annotation, obviously the usual stop words and so on. Then you see brief entry point, visual entry point to either tissues, organisms, or diseases as tagged in the metadata, and you can just see what are frequent data sets, and then you can sort of zoom into the less frequent ones. So this gives just a graphical way of navigating the data collection. And then on the right panel, to the right, on the rightmost panel, you see an overview of the participating repositories and their respective contributions. Blue, or here, this is PRIDE. And if you go around, you see the EGA, the European Genotype-Phenotype Archive, the Metabolome Workbench based in the US, Metabolites, EBI, Peptide Atlas, Seattle, and Massive at UCSD. And you see their respective contributions. You can also browse by omics types, because we currently have still a bug in the indexing. Genomics is not properly listed here, but this will be sorted out with an update of the XML. Moving on to the next level, we get a more zoomed in view of the data sets. One panel shows the latest additions to the data sets. The next one, the middle panel, shows the most accessed data sets. And then there's again a statistics panel, which will probably replace with another more data set centric view. As you can see, we use consistently these little green P's or green M's to mark proteomics, metabolomics, and blue G will be genomics, so that at one glance you can see the technology. In the last panel, you see a new section, which is a rendering of the omics Twitter account. And then you see, so we can always disseminate news via Twitter, and they are also shown on the website. And you have a statistics overview and the standard information about the project. So now, going into the top search bar, I'll search for, let's say, mitochondrion, if we can type properly. Mitochondria, so you see the auto-completion, and then you get the real search results of the available data sets. And you have the usual navigation paradigms. You can use the faceted search. You can narrow down by technology, by repository, by organism, tissue, and of course this list of criteria which the faceted search provides can be extended easily. If you zoom in on one particular data set, let's say this one here, then you get into the actual data set annotation. So this is really what is provided by the source repositories as the metadata for the specific data set. And you can then, from there, access the actual data in the repository. So again, we are only centralizing the metadata. For access to the real data, you then go with links, which will, of course, in the long run be harmonized. You can access the actual data for each in the repository for each of the data sets. We also provide for the publications which are listed as being associated with the data set. We provide a nice rendering of the publication, including altmetrics, to give you an indication of how heavily this publication has been referenced. Again, not by the typical reference count, but by the more sophisticated altmetrics system. And Then to the right of the render, you see two features which are so far very experimental, namely similar data sets. I think this is a very valuable and interesting addition in the long run, but currently we are only using the standard SOLAR mechanism to identify similar data sets. So at the moment you will see some way you say, why the heck is this considered similar? So this is clearly something where we will need to do a lot of fine-tuning over time. And that's really the overview of what the system can do right now. And I will briefly go back to the presentation. And I've already mentioned next activities are similar data sets and ranking. So we also still need to invest a lot of time into ranking, into giving the most relevant data sets first, which, as you all know, once you have growing data sets, is a tricky business and very demanding. And the other thing where we are at the moment blissfully unaware of ontologies, and it's very clear that we have to bring in ontologies into the whole system. And obviously we have experience with doing that, but we haven't done this in the first iteration of the project. Actually, we have now done quite a lot of work on that in the background, but that's not yet in this demo version. And I would just like, because it's often a question, briefly give a view of the system architecture in the background. Again, this is using the existing EBI infrastructure, which indexes the entire EBI. So this is a very heavyweight infrastructure, but it uses almost exclusively public source code like Lucene, Solr, of course, the usual scripting. The one thing which is significant, which is a significant component, which is commercial, is LSF. We're using LSF for the job scheduling of, but that can be replaced by open source free versions if necessary. And of course, for the actual usage of the omics DI, only a fraction of the resources listed on this slide are relevant. So this is only to give an overview of what's there in the background, but which is indexing the whole EBI, which also means it has backup and everything and a high security level. And with that, I'd like to thank everybody involved, in particular the two developers, Yasser Perez-Riverol and Ming Bai, the entire Pride team, the metabolomics and EGA team. All of these three have done a lot to contribute the data. The web development team has provided adaptations to the existing infrastructure to help us. And of course, I'd like to thank NIH for funding this project and PayPay for involving us in it. Thank you. Questions?
Henning, this is Ron Margolis. This was a real tour de force. I have two questions. One, earlier on you mentioned that there's a validation layer between the data set and the XML. And I think validation of the data is very, very important. I'm just curious how you did that, who does it. My second question, I think, is a little bit more at a higher level, and that is, given that BD2K is also funding the development of a data discovery index, and that's BioCaddy, how much overlap is there, and how much can each of these projects contribute to each other, and at some point, do they need to be merged?
Okay, so I'll start with the second question. There is actually a kind of parallel history between BioCaddy and the omics DI. We had put the project more or less as I have described it here into the original BD2K UCLA Center application, and we were not aware at the time of BioCaddy, and we met each other and each other's intentions at the first BD2K meeting. And more or less at the same time or shortly afterwards, then there was also a joint NIH BD2K Elixir meeting at Heathrow Airport, where we discussed again this project. And we decided, so we have been aware of each other. I have given also a webinar already in spring to BioCaddy in their webinar series on proteomics change, which is kind of the architecture or one of the roots on which this project is based. And we are in discussion with BioCaddy, but BioCaddy is doing basically the very systematic, big approach and for a very broad coverage, while our aim was to get something going reasonably fast as a pilot in a limited domain, namely only the omics. And it's clear that we will need to work together to not have two independent systems, but eventually one system which really works. And how that will work out is still fairly open. We have a joint application with the BioCaddy team for an administrative supplement, which I understand has good chances, but I haven't heard anything official yet. And we are working, and the content of that would be to jointly develop further what we have started here. And one of the reasons why we haven't done anything on ontologies is because we are very well aware that this is one of the things which are being developed in BioCaddy. The same thing, we have not really done a lot of work to structure the metadata, and BioCaddy is doing a very systematic collection of metadata formats of different providers. And so we have tried to really focus on getting something going for our limited domain, and eventually we will integrate with BioCaddy, and we will have to achieve one working system. But on the other hand, we needed, for us, this is only one step in a wider set. I took out that slide, in a wider workflow where we need to go from data discovery via data access to data analysis within the project runtime of a bit more than 3 years. So that's why we wanted to get going quickly. So that was, but you might also have seen on one of the slides I mentioned the sort of stakeholder timing. And again, there's a BioCaddy webinar planned in a couple of days where I guess we will start discussing in more detail how we go forward together. Coming back to the first question, the validation at the moment is very straightforward. We are just validating based on the list which I mentioned of mandatory and desirable fields, whether there is content available in these fields. We are not trying to validate the actual content. Again, we haven't added ontologies into the mix yet. We don't maintain allowed value sets for each of these fields. This is all something which will come over time. But this is already helping us to go faster in the development, because in the integration of especially the EGA, which had a significantly different metadata model, there was a lot of back and forth, and so the validator has been developed reflecting this. Okay, so to repeat the question was, what is the relationship between the existing EBI indexing system and the omics DI? And the answer is that we are using as a backend the existing EBI indexing system. So we have just adopted the strategy which has been in production at the EBI for years, actually. And this is just, we have worked with the EBI web team. They have adjusted their web services a bit for us. But we are using the same infrastructure, the same approach as the existing EBI indexing system. And this is just one outlet, basically one view on the data, currently this view only shows the omics DI data, and on the other hand, the EBI indexing system doesn't show the omics DI data at all, but it would be half a day's work to show, as I mentioned, for example, we could with very little effort, we could directly from the omics DI, for example, link to the molecules which are in the different data sets. We are actually for proteomics already doing this. I just didn't demonstrate it here. You can search for a proteome for a UniProt accession number, and you will get all the data sets which contain this accession number. And then it would be also very easy to then link to UniProt or to, let's say, Reactome or Intact to show this protein in context if we wanted. But we haven't done that at the moment because we wanted to keep focus. But we are really using the same system. And the technical, that's also why I showed the technical slide and the very big resources on the technical level. These are the resources used to index the entire EBI. Obviously, to index 5,000 data sets of the omics DI, you can probably do it on a laptop. But there is a lot of, well, infrastructure behind which provides security, but also it's clear that we only need relatively limited resources, and as I mentioned, it should be fairly straightforward if we want to extract the omics DI from the existing EBI system. It's all open source except LSF, and so it should be fairly straightforward to install this somewhere else. So the question from Ian 4 was, again, going deeper into the relationship with BioCaddy and whether what are the plans, what are the concepts of working together on the metadata structure, so, and also the scope of the respective projects. It's clear BioCaddy has a mandate to be very broad. We have a limited scope on omics. And now coming to the answer, I think we are, again, we wanted to get something going quickly based on the currently collected metadata, which is available in the participating repositories. And BioCaddy is using a much more structured long-term approach. And I think we will go along with the more structured approach and will hopefully be able to relatively agile adjust what we are doing to the more structured approach of BioCaddy, and will make, for example, and this is just now really associating, make available the XML files which we are using anyway in a way which is compatible with BioCaddy requirements and taking the federated approach to the next level. So I believe in federated approaches in general, and we have taken a federated approach here. I don't see why be able to adjust that a bit to make to fulfill BioCaddy requirements where there might be a much larger federation. If this is where things are going, it might also be the other way around, that we integrate that BioCaddy helps to generate compatible representations of data for other repositories. So we really have to figure out how we can best interact and coordinate what we are doing, but I think federation and integration on a relatively thin level is where I think we can have an impact rather than trying to be perfect and working forever. Take my notes.
My name is Wei Wang. I'm a professor of computer science at here at UCLA. I'm also the director of the Scalable Analytics Institute here at UCLA. And today I'm going to first give you a brief overview of ASTAC. We gave this presentation yesterday, but just for the sake of completeness, I'll give you a brief overview about the architecture design of ASTAC. This is about 2 months young project, so everything is pretty much in progress. So for ASTAC is representing A to Z technology, we hope to build a resource discovery index that include 4 main sources of BD2K resources. And the first one are the actually the outcome, the deliverables from all the BD2K efforts sponsored by NIH, which including not only the 11 centers, but also the Data Discovery Index program, the training program, and the targeted software development program. And the second category would be that published in literatures. For this category, we plan to doing extensive text mining and then try to accomplish this as automated as possible to actually release the burden from the author or the developer of those resources. The third category would be the by author submission. This could be either author initiated submission or is invited by SDAC, by us. And the last category actually is the one that we are currently focusing on would be to create a federated global resources that including all current available public repositories. And here you see on the screen would be a subset of those. So far we have been able to import about 4.5 of them, including Galaxy, BioGS, BioConductor, BioCatalog, and Elixir. So this is an overview about the set of metadata. Actually, this is still a subset of the metadata that we are currently keeping track of for each of the resource we index. The first column would give you the name of the field that of the metadata we keep track of, and together with the next column of the type of this attribute and whether or not it's mandatory on the third column indicated, and then the last column give you an example value of the metadata. As you can see that we intend to use the DOI as the public ID to identify each resources. We will also use the ontology EDAM to actually provide as definition of the functions and the domain categories and input and output format and so on. So we will also keep track of the author information, where this resources is published, and upstream, downstream tools, and the implementation platform, and so on, and the release date and update date. So in terms of the identifier and the ontologies we plan to use, that includes the EDAM that I just mentioned, and also for authors and the publications, we're going to use ORC ID for author ID and the DOI for publications. And the same thing for the resources. We plan to give each new resource a new DOI if there's no pre-existing DOI associated with that too. If there is one, we're going to just use that one. So this is the overall architecture design and the workflow. As you can see that we indicate the 4 main sources of data that we plan to incorporate. And we also plan to provide a RESTful API and for the to interact with the user. And the backend database would be MySQL, Solr, MongoDB, and Neo4j. So here are the current set of technologies that we are using for our prototype. At the backend, again, I said that we use MySQL to maintain the data integrity, and Solr to enable the scalable search, and MongoDB to provide us to track the search statistics and the resource statistics, Neo4j for graphical representation of the workflow. And we plan to deploy it on Amazon Cloud pretty soon. Right now, the development of the prototype is on a local server, but we plan to release that pretty soon. And on the front-end interface, as I said, we're going to provide a RESTful API And for all the visualization, we use these three to provide that. And in general, we are using Node.js and MVC design pattern throughout the development. Now I'm going to give you a live demo of this tool. The current URL is www.adstack.bio if you're interested. And then first, you can see the screen that at the top, right, we have the home button, the advanced search, register, new resources, and the middle is a logo, and then the bottom you would see a search bar, and which would enable us to do a simple search, either by name or substring of the names, DOI, description, tags, or author. And for example, let's try something very simple. Let's see, variant. Okay, I would expect a lot of resources that actually match well with this. As you can see, a long list, right? And then for each of the two, and you can see that actually we have a short description and a summary of the metadata, and we have the tags. And then the left part, that's you see that's enable facet search. There's we have this like inside the square, this letter indicating the type of the tools, and the domain, the platform, and then the language, and then the sources. And then let's see if we're interested in the first one. You can click here, and then that gives you more detailed metadata, and then which also include the links where you can find the source code, the homepage of the project, and then the documentation associated with that. And if you click this publication, the DOI here, it will send you directly to the papers page. And this particular one is published in Genetics. Okay. So let's go back. Let's see, let's do another search. Let's see, we're interested in some genomics tools of RNA, for example. And then, but I don't have a very specific idea what the name of the tool, so I just search RNA. As you can see, this is a very generic term which returns us a lot of result. And then, but I know I'm interested in tools, not database, not widgets, so that I could just click on the tools, and then that's helped me to narrow down the search a little bit. But still, I have 235. And then I said, well, I know that I'm interested in the domain of genomics. I could just click genomics here. Then that's helped me to narrow down to 189. And then I said, well, maybe I want to find something that implemented in a language that I'm familiar with. Let's see, C++. That's giving me 3. As you could probably also notice that on the right-hand side, it's give you the search history. This is RNA is what I type in, and then this the sequence of refinement that I just did. Okay, and let's see. Well, out of these 3, I'm interested in the top one, top hat 2, and this is a RNA-seq alignment method. Again, you can see the one that similar to the one that I showed you on Caveat. And then you can see that actually on the right-hand side, you notice that we said coming soon because it's not fully functioning yet. We hardware many of this just to demonstrate what we intend to have. This is a pipeline that Top Hat 2 is part of. And we are going to have a reference data set, suggest you some sample data set that you could run this. And also, for example, once we look at this Top Hat 2, and then we may realize that, well, we're not sure whether this is the one that we want to use, so we want to look for some alternatives. And then we also recommend similar resources. For example, this one is Selfish. You can just click it, and then it's give you to the page of the Selfish. Okay? And then, So that's, you can play with this, and then let's go back to do another search. Again, something general. I want to do some alignment. Let's see, RNA sequence. Okay, again, it's pretty general. And then I realize that's too general. And then what I can do is that I would be able to further refine it by doing the advanced search called sub-search. And then if I do aligner here, that can help me to narrow down things. Okay? And then maybe I realize, well, that's too restrict. Maybe I don't want to restrict myself to RNA sequence. Then I can just select some of the one that I already did, and then let's see, I just want to remove RNA sequence and keep the aligner. Then this is just result. It's now 19 queries. And then, so, and alternatively, what I could do if I go back to the home page, and then again I do RNA, there's other ways I could do would be, for example, there. I still do sub-search, but then you can see there's filters already built there for tools. And then perhaps I want to do the language. Okay, I want something implemented, let's see, in C++. I just put in C++, and then that's give me all the things implemented in C++. And then you can see that actually these are, refined to 5. I can just, click some of the tool. Let's see, TopPy tool. That's what we did before. And I think at any time on the right-hand side, it gives you search history. You can add it to additional terms, remove some of the terms, and then go back. And then on the left-hand side, those are the facet search that are enabled. And then you can also maybe, click the text. It will also give you that additional quantification. So that's something you can click. Let me see. Going back. So then we can play all day long with this, but then what I still want to show you would be that if you go down on this front page, as you can see that we also keep some statistics, like the top resources, like the overall statistics of the current repository. You probably noticed that, again, from yesterday it was 5,000 something, right? Some resources, now we have 400 more since yesterday. And then there's some visualization of the distribution of the platforms, tags, the repository we have. And then, for example, you can also go, if you're interested in something implemented on Linux, you can just click that. It's supposed to give you all the tools that could run on Linux. Okay, and then you can continue to do the search as we did before, and then you could, for example, click the tag genetics, and then it give you everything related to genetics. Okay, and then last thing I want to show is let's go back to the page, and you notice we have a register new resource button here, and it's give you right now. It's give you a simple interface. You could manually enter any resource, and it's functioning now, but it's just not as pretty as we hoped. So the layout, everything, we're still working on that. Even though you can notice from our poster to yesterday's presentation, even for today's demo, for some of the pages the layout is different because we are continuing improving it. So, and also we appreciate any suggestions and feedback. Thank you.
Hi, this is Heidi Sophia at NHGRI, and are you accepting and encouraging new registrations of new resources? Because I have a whole bunch that I would love to see registered.
Yes, we love that. Yeah.
That's fantastic.
Yeah. Right now, because this is not made public yet, so we don't have the real statistics to show on that. Right now, all the results are ranked just by similarity to the query. so far. Yeah. What we plan to do is that whenever there's, it's especially commonly used tool. Let's see, a good example of this would be Top Hat versus Top Hat 2, right? They did change a few things, and sometimes like what used to work no longer work anymore. And what we plan to do is that actually we're going to do intensive text mining of the product website, of the publication, of the blog, right? What user complain about, and then so that we can keep that up to date. Of course, we will send to the author for validation to see whether they agree with what we summarized. So we intend to have the combination for automated analysis versus and the human curation to make sure that we can keep track and everything and make sure the workload is manageable.
This is Dawei Lin from NIAID, and I have two questions. And one is what's your plan to update the entries already in your database? And the other one is that how do you classify programming languages? Because some of the tools may use a mix of languages.
Okay, so that's two questions. The first question was how do we keep up to date to the changes that we're going to use the same method as I just described for those major ones. So for the minor ones, we will basically keep track of when was the last time they update anything, release anything, maybe small bug fix, and we will basically as part of our metadata, and we will show that when was the last update. And because we are not going to actually physically store all those tools, we only keep track of the metadata, and then you can always go back to the website of the original project, and then they will have additional information for that. The second question, what was the second question? Can you remind me? Oh, multiple languages. So we have, even though so far the one we indexed, most of them just were implementing in a single language, but yes, especially in the pipeline that different tools may implement in a language. In this case, actually, I think we are allowed the for that field, you can have multiple selections there. So it's basically if you say, well, it's implemented in Python and R and C++, then we'll have all indication there. So it's not just the one language that we're allowed to be listed there. It could be a set of languages. Thank you.